as well as some updates on how to claim CME if that is what you're doing today. So just for the numbers, the UDN has now received about 7,500 applications and accepted about 3,000 participants. We've completed evaluations for about 2,650 participants. And at this moment, we have diagnosed 826 participants. And our mission really is to improve the way that we diagnose and care for patients with undiagnosed diseases, facilitate research into what is causing these undiagnosed conditions, and to create an integrated and collaborative research community, which I think is really highlighted by these grand rounds presentations. Just to orient you a little bit to where we are across the country at the moment. So we have clinical sites, research cores, the Data Management Coordinating Center, and the Sequencing Core, which we'll be hearing from today. And then not going to go through all of the institutions, but the UDN continues to grow. We added new sites to the UDN in 2024 and will continue to do so moving forward, which we're thrilled about. So for our policies for grand rounds, we welcome you to ask questions via the Q&A text box. We'll get to those at the end of the presentation. And then the content in today's presentation is provided for informational purposes only. So intended for a target audience of multidisciplinary healthcare professionals and students, and information may be used for personal use if the content and references remain unchanged, and no part of the presentation can be published without prior written permission from the UDN. All right, now we are thrilled to introduce Pengfei Liu, who is an associate professor, director of the Medical Genetics Multi-Omics Laboratory, director of the Laboratory Genetics and Genomics Training Program, and in the Department of Molecular and Human Genetics at Baylor College of Medicine. So with that, I will turn it over to Pengfei to get started. Thank you, everybody.
All right. Hi, everyone. Today, we are excited to share some of the experience we learned from implementing clinical transcriptome RNA sequencing as a clinical test within the UDN Undiagnosed Diseases Network. So RNA sequencing has today, the type of RNA sequencing that we're going to talk about is focusing on the diagnostic aspects, right? So the goal of applying whole transcriptome RNA sequencing for these patients with Mendelian disorder is that we want to facilitate the molecular diagnosis where the foundation of the molecular diagnosis is built on DNA sequencing. And we hope that RNA sequencing can provide high throughput and effective data to help us improve this process and increase the diagnostic yield. So why are we doing RNA sequencing? I want to make an analogy here, right? Consider you have the potential to do both DNA sequencing and RNA sequencing to find the mutation that caused the patient's disease phenotype. The analogy is that, can you find the mutation in this score note, right? Can you do that if you have the DNA sequencing, which is presenting on the screen? You might be able to do it if you're proficient in music, but might be tricky for many of us. But if you can express this sequence out and hear it, just like when you have RNA sequencing, it is made apparently easier. I hope that you heard the play of this note and appreciate that there's something wrong here, right? So hopefully this illustrates that if you have a tool that can express out what is the actual consequence of the mutation, which is done in RNA sequencing, this really makes it easier when you interpret the data and when you're trying to find the mutation to the patients. So RNA sequencing, here is a summary of what has been published in the literature from research groups and translational efforts using RNA-seq to solve different disorders. And the first thing I want you to note is that different tissue types can be used, primarily the clinically accessible types, blood, skin fibroblasts, and sometimes muscle biopsy. And they can be used for different types of disorders, either unselected genetic disorders with various type of diseases, or you may have specific disease types, such as muscular disorder. When you have muscle, you obviously have a higher diagnostic rate. And some other times, when you know that your target disease has a higher expression, has a higher gene expression representation in the tissue type. For example, skeletal disorder, a lot of the genes are well expressed in the fibroblast. You may choose fibroblast and achieve a relatively higher diagnostic rate. But overall, these studies have shown that we can achieve from 7-8% to all the way to almost 40% diagnostic rate using RNA-seq. So we started applying RNA-seq as a clinical test at last year, 2024, from the Baylor sequencing core. And this is also the first time that we tried to clinically validate a whole transcriptome sequencing test and use a outlier-based analysis approach to try to implement this in a clinical workflow. So we've been taking skin fibroblast samples or blood samples and processing them in two separate wet lab processes. And here are the description of the bioinformatic pipelines. And then finally, as I said, we use outlier-based analysis to try to identify the candidate pathogenic variants, which I'm going to show you more in the later slides to give you actual examples. So in this process, it is critical that we have the DNA data, which will really make the interpretation of RNA sequencing much more effective. Also, this work has been summarized on a preprint and is currently under revision, under peer review. Hopefully, we'll publish the work soon for you to see the validation. So before going into the validation, I wanted to share some of the upcoming changes, some of the updates we want to make to UDN sample submission and sample type selection. So you already, I know many of the audience are from UDN clinical sites, you are already submitting samples to us, but we are planning to make some more refinements to define the sample types, right? So we are already collecting blood in packaging tubes and live cultured fibroblasts or cryopreserved fibroblasts, but we're gonna have open this new sample type skin biopsy. So we will be able to start from skin biopsy very soon, and we will send an update when everything is finalized. Previously, we were accepting extracted RNA if they were extracted in a clear environment, but after some experience, we think that it will be better to control the variability if we do all the RNA extraction here in the sequencing core. So we will propose that in the future, after we share these instructions, these updates, we will no longer accept extracted RNA. We will really prefer blood or fibroblast or skin biopsy samples. However, we understand that in this particular UDN project, which is a hybrid between research, investigation, and clinical implementation, there may be situations where we only have extracted RNA, and there's not a feasible way to get other sample types. So in that situation, we will still accept RNA, but convert the report from a research report, even though we will put this through the same wet bench and analytical workflow. But on the report, we will just say that it is called a research report because it has not gone through the same standards as we did through the validation of other clinical samples. Additionally, one important lesson we learned is that it is really, really helpful to have the DNA data when we interpret the RNA data. I'll show you some of the data supporting this in the validation, from the validation work later. So we want to remind everyone, and we will have this instruction in the updated version, that please upload the DNA sequencing data if they are not already in the gateway. If they are from another research effort or from previous clinical testing, we will really appreciate having that uploaded to the gateway. We are able to analyze the RNA data without the DNA data, but it will be less effective. So coming back to the validation, before we start the validation, we wanted to make some definitions. There are two key analytical elements that we were focusing on in RNA sequencing data: expression level and splicing patterns. These basic elements are analogous to the things that we are familiar in DNA sequencing, which is variants, single nucleotide variants, indels, CNVs. We don't have that. We're not focusing on that in the transcriptome sequencing data, but expression level and splicing pattern. So what do I mean by that? Here are some basic examples. Expression level is can be reflected by the read counts that are obtained from the sequencing data over the gene. So here's an example of a gene, and you can appreciate that in the control sample, there are 6,000 reads, and in the patient sample, there are about half the read. So this is suggesting that the patient has reduced expression in that gene. What do I mean by the splicing pattern? So the splicing pattern can be defined by percent of reads at the junction, at each of the junctions in the transcript, supporting the particular splicing pattern. So here's another example. If you look at the control first, the sashimi plot demonstrates how the reads are supporting the splicing. But in the patient data, you see a different junction. So in the control, if you focus on this junction, 100% of the reads are supporting the canonical splicing. But in the patient data, some of the reads, about half of them, are split to support an alternative type of splicing, skipping that exon, right? So we can use metrics such as PSI, percent splice in, to quantify this and measure whether the patient data are significantly different from the control data. So in our validation work, one of the first challenge we had is that we don't have as good as a benchmark as we have from DNA sequencing. So we have to generate one. We used the genome in a bottle data and generated a provisional benchmark defining what do you expect to see in terms of gene expression and splicing. So we were able to use this benchmark, and we sequenced the genome in a bottle sample in a couple of repeats, and we were able to measure the sensitivity, specificity, all of these metrics that are required for a clinical validation test by CAP and CLIA. Similarly, we did that for reproducibility, and we did it for both the genome in a bottle sample and some other publicly available, widely available cell lines. So next, we defined reference ranges. This is a concept that is not very much investigated in DNA sequencing, but is very critical for RNA sequencing. What are we looking at for reference ranges? Right, so I'll give you some example of how do you appreciate the reference range for expression of a specific gene and splicing of a specific junction. So looking at gene expression level first, here is an example gene LZTR1. And if you measure the expression level, which is reflected by the number of read counts in this gene across a couple of control samples we have sequenced, we see that most of them, they are scattered around 1,500 to 2,000 reads. So that is kind of defining the normal range. you can make a statistical range for that and have an upper boundary and lower boundary based on your control cohort. Here is another gene, DNM1, and you can see the spread is much wider and also reflected by the lower count. So this gene is actually not well expressed in blood. So that means we have a very wide reference range, which means we will have much weaker sensitivity to detect clinically meaningful changes at a smaller scale, right? So first we plot the what I mentioned, the lower and the upper fold change cutoffs for all of these genes, for the previous gene LZTR1, and now we plot that for this current gene, DNM1, and you continue to do this for many, many genes, for all the genes in your transcriptome, and you get this distribution of the reportable range, the lower reportable range and the upper reportable range. And you have a graph like this for both fibroblast and for blood. And of course, you can also pull individual gene data when you are analyzing your transcriptome sequencing results. I wanted to emphasize that this is providing us a reference range, but is in no means saying that our test is specifically sensitive at a particular expectation for each gene, right? That if you want to get that data, you will have to analyze the gene specifically and generate gene-specific validation data, which is very difficult, but at least we have a reference for us to refer to. Similarly, you can establish the reference range for splicing. This is an example for the gene PRUNE1. You can appreciate the reference range for all the exons. Some of them we have perhaps have more reads, so you can have more sensitive reference detection compared to other exons. So after establishing the reference ranges, we moved next to testing clinically positive samples. These are samples that we know there are RNA findings, abnormal RNA findings, most of them previously identified in the UDN collection supported by various research sequencing data. And we challenged our sequencing test to see if we can detect these abnormalities as outliers. And by saying outlier, there are two types of outliers. First, expression outlier. If the expression of that gene is significantly lower or higher compared to the control reference range, or if the splicing stands out, right, it's something that's not observed in the control reference range, they will all be labeled. So here's a summary of what we detected in the validation cohort. There are different colors, and you can first look at the red color. They mean that these findings are very challenging. They actually weren't detected. And almost all of these are because the gene has low expression in the sample type. Right? So we have no way to detect them. And the green and the yellow colors, these two indicate that the finding was identified, but at different significance level. Right? The green ones, means that they are detected under very stringent criteria, and the yellow versions indicate that they are detected at a looser significance level. So why do we have two different significance cutoffs? This is because on the one hand, we wanted to detect these positive findings as much as possible, which means on the y-axis, we want the numbers to be high. But on the other hand, we don't want to have too many variants to review, too many false positive, non-diagnostic variants. You can see the example here. If you want to detect all the positive findings at that kind of significance level, you have to review over 1,000 variants, outliers per sample. So we have to strike a balance. And we have this more stringent criteria where we are going to miss some of these positive findings, but it is only really giving us manageable number of samples, outliers to review per case. So to summarize this finding in the next slide, we have the two analysis mode. One is the targeted analysis mode. This is analogous to when you have the DNA sequencing data and when you know where to look at in your RNA sequencing data. So you don't have to have that much of an extremely sensitive significance value. So under this analysis mode, you are able to detect most of these positive findings. And you only need to review the candidate regions that you are suspecting based on the DNA data. On the second agnostic analysis mode, when you don't have DNA data to guide you, or when you are looking beyond the DNA data, something that may be missed from the DNA sequencing, we can still do the analysis. We can still manage the number of outliers to a few dozens per case, which is feasible for us to review in the clinical workflow. But you see that we're losing some of the sensitivity to detect those more challenging or those outlier events with less dramatic changing effects. So what we learned from the validation is that we should combine these two analysis modes in our actual clinical, routine clinical work, meaning that we will always do this agnostic analysis mode, and we will have a better chance to identify those more dramatic changes. But on the other hand, we really would like to have the DNA data to guide our analysis to the targeted regions. So you don't need to achieve extreme genome-wide significance p-value, but you can still identify those outlier findings suggested by the DNA data. So now I'm going to talk about our actual clinical data. We have just started transcriptome sequencing for a few months, and we really don't have a lot of sample volume. But based on the current volume, we have about 10% diagnostic rate. And here are some of the positive findings from those analyses. I'm going to present two of them today. This first case is from the NIH sequencing, from the NIH clinical site. This is a 19-year-old male with cerebral atrophy and hypoplasia, impaired balance and wide-based gait, diplopia, childhood onset pernicious anemia, and hypogonadotrophic hypogonadism with sarcopenia and hypospadias. Genome sequencing was done in the Baylor sequencing core, and a compound heterozygous variant were reported in the PRDX3 gene. When this was initially reported, the gene was not known to be associated with diseases, but later, soon later, several publications reported that spinocerebellar ataxia autosomal recessive type 32 is actually associated with this condition, which matches the patient's phenotype. So these are good, very promising variants. And one of them here is a splicing variant, +1 splicing. So RNA-seq was ordered, trying to understand what is really the consequence of this +1 splicing variant, and what can we classify this variant as. Before going into the RNA-seq data, I wanted to show you the prediction, the computational prediction from Splice AI. So this is where the variant is, and Splice AI is predicting that there is going to be gain of a splice donor, and there is going to be a disruption of the canonical splice donor. So meaning that the exon is going to be extended a little bit into the intron. This is the prediction from Splice AI. So after performing the RNA-seq data, first, we see that this gene is labeled as an outlier with a p-value. It's not very significant, and you can see that's because the fold change is not very dramatically reduced. This is 80% compared to the controls with a z-score of 2.56. So here is I'm plotting the TPM values of this sample compared to all the other samples. If you just focus on the fibroblast, and these are data from the blood, which are not directly comparable. And this patient is indeed on the lower end, but is not to the scale that makes it really stand out. But it is still significant. This is what the splicing data show from the RNA-seq data. First, I'd like you to focus on the control data. And this is where the mutation is, the variant is. The control data show that most of the reads, almost 100% of the reads, are supporting this canonical splicing pattern. But in the patient, there are about 83% of the reads supporting this canonical splicing. There are alternative reads that are supporting different splicing patterns, and they add up to about 14%. So this is how you read, if you read the raw data from the splicing outlier analysis, it will say delta percent splice in is 14%. This is what it is suggesting. Zooming into the actual splicing pattern is extremely complex. So first, there are 15 reads supporting the canonical splice, supporting a cryptic splice donor, right? There are 15 reads. This is what was predicted from Splice AI. In addition to that, it's much more complex than the prediction. There are 9 reads that support going through the reading through into the intron. So the entire intron might be retained to some degree. There are 8 reads supporting skipping of the exon, right? So these 8 reads are skipping this exon. And there are additional different cryptic splicing, abnormal splicing patterns that I'm not gonna go through. So, but hopefully you can appreciate this is a really complex splicing consequence. And each of them, because we are doing this laboratory testing, we are supposed to describe them in a precise nomenclature to document what was observed in the transcriptome data. And here they are. This is the first one, which is what was predicted by Splice AI, insertion of some sequences in the intronic region. And here are the additional nomenclatures. I'm not listing all of those because I think that will certainly be overwhelming. So on the report, we included this abnormal expression and abnormal splicing results. But we thought it would be helpful to simplify that, to really summarize what is the take-home message instead of showing all of these different versions of complex nomenclature. So for the splicing, we just concluded that there is a complex pattern and there is many different abnormal splicing reads, and we are seeing low fraction of a lot of them, possibly because nonsense mediated decay. And we also highlighted that there is this observation of reduced gene expression. Gene expression detection is always challenging to say we saw 80% compared to the reference, but does this patient really have precisely 80% of the gene expression? It's always tricky to make that conclusion, especially when you combine this observation with the complex splicing consequence, as well as another observation which we looked at the allele in compound heterozygous to this variant, and we assessed how much percentage does that allele in trans account in the reading sequence in the sequencing data. We observed that the allele in trans has a much higher fraction than 50%. So this is suggesting that the allele with this splicing variant has undergone significant nonsense mediated decay. So instead of giving a precise number of what percent of gene expression is, we decided to present all of these raw data and give an overall impression that there is reduced gene expression because of the complex splicing patterns. So that was the first case. And the second case is from the Baylor sequencing core. This patient has, in summary, recurrent spontaneous cervical artery dissection. This is a 2-year-old female from monozygotic twin, initially prenatally with prematurity and twin-twin transfusion. The patient was developmentally typical to begin with, and until 25 months of age, there was an illness of cough and low-grade fever. And after that is resolved, the patient started to refuse to walk and showed focal weakness and then had a seizure. Brain MRI was done, which showed multifocal subacute and chronic infarcts. And also there were dissection of bilateral cervical SCA and right vertebral artery detected. Here are some of the imaging results from MRI showing the acute and subacute infarcts. Additional imaging showing the subacute and chronic infarcts. This is on the left side, SCA showing the occlusion and dissection. This is on the right side, occlusion and dissection. Here are the vertebral occlusion and dissections, also in the same patient. Additional imaging results. So imaging of the twin sister was performed. And even though there were no evidence of any infarcts, but we observed tortuous vessels and duplicated left vertebral artery. We didn't perform, we haven't obtained genetic data of the twin sister yet at the moment. Family history is negative other than the observations from the twin sister. So initially, whole genome sequencing was performed, and some variants of unknown significance were reported. They're all intronic variants in COL4A2 and also in PDCD10. And there are deep intronic variants. Because of the potential phenotypic relatedness of these genes to the patient, and also the low population frequency, there was a reasonable suspicion that these variants could be contributing. So we wanted to use RNA-seq to evaluate these targeted events, which we did. And from the RNA-seq data, this was sequencing performed on fibroblasts, and many of these genes are expressed very well in the fibroblast. We saw no abnormal splicing and no abnormal expression levels. So the data, even though we cannot say that the two variants are excluded from suspicion, but these data, they do not provide additional support for the pathogenicity. So making us to look at other regions of the genome, of the transcriptome. We did find something else that's suspicious from the gene RASA1, RASA1. This is a gene that's related to capillary malformation, arteriovenous malformation. so CMAVM. And the gene may not be a perfect match, but it is indeed related to the phenotypic presentation. This is a deep intronic variant, 200 nucleotides into the intron, and we detected some abnormalities from the RNA sequencing data, which I'm going to show. So first, I will present the computational predictions from Splice AI. Splice AI predicted that there is 0.5, around 0.5 score of acceptor gain associated with this variant. This is where the variant is, right? And there is some possibility of a cryptic splice acceptor generated here. And with that as the only predicted aberration, the expected outcome is that these long intronic sequences can now be incorporated into that exon, right? Making it longer, right? So the original sequence between the previous exon and the current exon, we are inserting a good chunk of new sequences. This is an alternative way to list out all the actual sequences, or almost 200 nucleotides. And based on the prediction, this is going to result in a frame shift. And this is in the middle of the gene, so we expect that there should be nonsense-mediated decay. So we got the RNA-seq data from the transcriptome analysis, and indeed, there are some pretty significant abnormal splicing events detected. There are two major groups. I'm showing you the IGV raw data from the patient compared to the controls. And I'm highlighting the two groups of abnormal splicing consequences by different colors. The first one are indicated by this red. So these are junctions connecting the previous exon to somewhere in the middle of this exon. So it means that a chunk of this current exon is being spliced out, right? That's being removed. And this is reflected into this nomenclature here, that we have a short segment of deletion, and this will be out of frame. The second consequence are reflected by these blue colors. So we have inclusion of the intronic sequence, and I believe this is the same event that's predicted by Splice AI. Here it is, the same nomenclature as I previously shown in the last slide. And you can see similar level of intensity supporting these two abnormal junctions. And they are both pretty low compared to the reads supporting the canonical junction, which is around 200. And these two abnormal splice junctions are around 10. So how to put them on a report? This is getting more technical into the nomenclature. If you read the guidelines from the HGVS website, in a situation like this, where we have one DNA variant that resulted in a mixture of different consequences, right? You should describe them in this format, and you should list all the consequences within this bracket and separate them by comma, right? So these are the two different ways. Both of these are acceptable to describe what we observed in the RNA sequencing data, right? The two different consequences, the red and the blue that I showed you in the last slide. So expression outlier analysis for this patient, we actually didn't detect an outlier. This patient has an expression that's pretty much in the normal range compared to the other samples that we have ever run. This is the TPM and also the statistical analysis. The drop analysis didn't label this as an expression outlier. So one other way to strengthen or test this result is to look for SNPs, heterozygous SNPs, that can inform monoallelic expression, right? If you see monoallelic expression, like the last case, then you are more convinced that there is indeed expression reduction. But maybe because of the noise, maybe this sample has a higher baseline expression to begin with, but now it is reduced. It is still within the reference range. But unfortunately, we don't have informative SNPs for the monoallelic expression analysis. So even though we did not detect this outlier expression, I would say is non-conclusive in terms of the expression analysis. So on the report, we only reported the abnormal splicing, right? We described what was observed for the two aberrant splicing types, and we noted that abnormal splicing has not been detected as a statistical significant reduction in this gene. But we actually don't know, and this is really a challenging situation for us to make a conclusion of the clinical significance of this variant. Whether this is indeed contributing to the phenotypic presentation in the patient. So that's all that I want to show today. So today, as a summary, I presented first the objective of doing transcriptome RNA sequencing to improve genetic diagnosis and what is the current status in the field of using RNA-seq. And then I presented some of our data from the clinical validation and followed by 2 case presentations to show you really the analytical process and the rationale and the thinking rationale behind the scene. What do we do in the sequencing core when we look at this data and when we put this on the clinical report? And I would really appreciate feedback and suggestions. This is definitely a learning process. This is a new test that we are trying to learn, and we will really value feedback from all the clinical sites, from all the collaborators, on what we should put on the reports, what type of information are useful, what others might be misleading, and improve the utility of this test in a collaborative manner. So this is everyone who have contributed to the work. The data is generated in the MGML, the medical genetics multi-omic laboratory, and together with Baylor Genetics. And the 2 cases that I presented, the first one is from NIH, Dr. Toro's patient, and the second one is from Baylor, Dr. Lanami's patient. Thank you all very much for your attention. I'm happy to answer questions.
Thank you, Pengfei. That was an excellent presentation, a great overview for anyone who's not as familiar, but then the deep dive into some really interesting cases. So we will take some questions now. We have 2 that have come in so far during the conversation. And then if anyone has additional questions, please put those in the Q&A on the bottom of your screen. You should see it. So the first question came in through the chat, so I want to start with that one. Are you getting a sense, Pengfei, of how many canonical splice mutations are not causing splicing disruption?
This is a good question. So based on prediction from Splice AI, I think we can already get a pretty good answer. A lot of the times, the splicing prediction are pretty accurate from the current state-of-art programs. When there are usually situations when there is indeed alternative splicing, abnormal splicing, right? But the resultant consequence might be inframe, right? It may just include or delete 3 nucleotides or 6 nucleotides into the reading frame. And then even though splicing wise is different, but it may or may not be pathogenic. So all of these can be validated from the transcriptome sequencing data. And we really need to gather more data to provide experimental evidence to answer this question. What is the percentage that is not disease causing? That's contrary to our conventional thinking.
Great, thank you. So this is a kind of within the UDN logistical question. Are you providing reports for the transcriptome sequencing yet in the gateway, or still only data? And at whatever point you're able to provide the reports, is that going to be able to be requested for previous transcriptome data?
Yeah, so all of the sequencing being submitted, I think from the middle of last year, from I think July, August, around that time, we are all issuing a clinical report. From that point on, all the new samples, we have a clinical report, and that should be uploaded to the UDN gateway when the analysis is completed. Regarding the samples that were sequenced prior to that time, we will not be able to issue a clinical report for those because they were run under a different pipeline that's different from the clinical validation. So they're not clinically comparable to our cohort of controls. So we have to rerun them if you really want a clinical report. We have to rerun the sample under the clinically controlled workflow to generate another report.
Great, thank you for clarifying that. And there's a question here about the controls being used. So great presentation, a question. Since the same control being used for all case samples and sequencing runs are different, I was wondering if you would remove the batch effect. So I think if you can speak a little bit to batch effect, yeah.
Yes, that is something that is ideal that we can do, considering batch effects. In the current bioinformatic workflow, we use the drop process, the drop programs that just integrate all the controls together. So from that point of view, we're not specifically integrating batch effect evaluation in the review. But beyond that, when we do our manual review, as you can see, we always plot these TPM with other samples in the same batch, and we look at metrics like that when we review individual cases, right? And it will be great one day that we also kind of systematically do that for all the cases. But indeed, that's very important. Batch effect in terms of sequencing, batch effect in terms of RNA extraction, batch effect in terms of cell culture. This is one of the reasons that we are proposing to have all the cell lines shipped to us instead of the instructed RNA. So we can control this. We can control the batch effect to a better degree.
Great, thank you. So this is a question about the validation, and if you can share a little bit about the decision process and rationale to validate RNA-seq as a standalone clinical test with its own lab report, rather than incorporating it as an additional component of the genome sequencing report.
I really advocate that both versions are provided clinically, and both of these are validated in the same process from us, right? So as I said, interpretation of the RNA-seq data will have higher yield if it's combined with the DNA sequencing data. But there will be situations where logistically DNA sequencing data may not be immediately accessible, right? Or it may not be in a format that that can be controlled in a clinical environment and analyzed with predicted sensitivity, specificity, etc. Right? So that's why we are having this alternative version where if you only have RNA-seq data, we can also do the analysis, but acknowledging that we will have lower detection sensitivity because of the lack of the DNA data. And one more thing we do with the DNA data is we always do identity check, right? Because this is always a concern in the clinical lab when you are, especially when you're culturing cells. What if there are sample swaps? With the DNA data, we can confirm that this is the right individual. And we do that for every sample as a quality control step if we can get the DNA data.
Great. That's really helpful to hear about. This is a question about the RASA1 variant. Was that variant in the child with dissections de novo? And if not, was there a positive family history? Do you know?
As far as I know, we don't have family member specimens, right? If the clinician is online, they may chime in, but from what I know, we don't have access to other family members' DNA data.
Okay,
So
and it looks like Jill Mokri from Baylor Clinical Site commented that testing is underway in family members for the RASA1 gene. Thank you, Jill. Thank you, Pengfei. Okay, I have one last question. I want to also, since I know we have the new CME process, I'll put the slide back on the screen so people can start to click this link before they have to get to their next thing. But one last question that's come in, Pengfei, is are there any efforts by ACMG, CAP, or others to come up with standards for RNA-seq analysis and reporting similar to the standards for exome and genome sequencing?
This is our hope that we gather some initial data so that efforts like this can be promoted. This is, I think there's clear evidence from the translational field that RNA-seq is very instrumental in improving the diagnosis. And there are RNA-seq performed at different various types. There are those targeted sequencing, there are those panel sequencing, capture sequencing, but this one, transcriptome, is one of the more technically complex one. And certainly we will benefit by forming a consensus and having recommendation from the ACMG. I don't think we have enough data to form a consensus at the moment, but we are reaching towards that goal.